Hello students, I am teacher Svetlana, and in our today's lecture we will learn about the cells of the immune systems, which are the lymphocytes and the antigen presenting cells. This is covered in the chapter Human Health and Diseases. So let's begin. Students, there are two main types of cells involved in the working of the immune system. They are the lymphocytes and the antigen presenting cells. Lymphocytes are the main cells of the immune system. They, like the other blood corpuscles, arise from the stem cell, the hemocytoblasts present in the liver of the fetus and in the bone marrow of an adult. Have a look at this image here. You can see here, this is the bone marrow. You have the hematopoietic stem cell. It divides into the myeloid progenitor cell and the lymphoid progenitor cells. These further give rise to our other blood corpuscles, that's the eosinophils, basophils, neutrophils, monocytes, erythrocytes, and here you have the lymphoid cells which give rise to your T cells and B cells. Some of them undergo differentiation in the gut-associated bursal lymphoid tissues, the tonsils, Peyer's patches, and are called as bursal or B lymphocytes. Others are differentiated in the thymus gland and are termed as T lymphocytes. The mature lymphocytes pass into the body fluids, that is the blood and lymph, and circulate in the body. Many of them stay in the lymph node. B lymphocytes form the humoral or the antibody-mediated immunity, and T lymphocyte forms the cell-mediated immunity. Have a look at this image here. This is the way your B cell appears, and this is how the T cell appears. Now, both of them perform different functions. So, your B lymphocytes produce antibodies, and your T lymphocytes provide you the cell-mediated immunity. Yes, students, I have put up a differentiation between the B lymphocytes and T lymphocytes. You can pause the video for a while and have a look at these differences. Now that students, we know we have two types of lymphocytes, that is the T lymphocytes and the B lymphocytes. Now we will have a look at the mechanism of their actions. Beginning with mechanism of response of T lymphocytes to antigens. Antigens, as I've explained earlier, it is the foreign particle or the disease-causing pathogen or virus that is entering in our body. Now, on coming in contact with an antigen, a T lymphocyte forms clones of T cells which are similar but they perform different functions. The clones has four types of T lymphocytes. The first one is the helper T cells. These sensitized helper T cells produce lymphokinins for performing several types of functions like proliferations of other T cells, that is sensitizing other T cells, stimulation of the B lymphocytes and the macrophages. Then we have the killer T cells or the cytotoxic T cells. These directly attack and destroy the invading microbes, infected body cells and cancer cells. Killer T cells bind to the infected cell and secrete perforins. Then perforins form a hole in the infected cell. It also releases substance that kills the cell, and hence the name cytotoxic T cell. Have a look at this image here. This is what happens when your T cell is activated. That is, a virus comes in, enters into the cell. We have an infected cell which comes in contact to the T cells. T cells gets activated, and this activated T cells, the initial one releases cytokinins which activate another T cells, which in turn leads to the replication or forming clones of this T cells. Now these clones are of different types performing different functions. One of them becomes the cytotoxic T cells, which bring about the destruction of the cell, and finally the destructed cell is phagocytized. Here you can see how the helper T cell on activation is doing its work further. That is, when a virus comes in and the helper T cell recognizes it as foreign, it sends signals. It sends signals to the cytotoxic T cells, and the cytotoxic T cells kills the infected cells. And on the other hand, it also sends signals to stimulate the B cells so that the B cells are activated and they produce antibodies against the invading virus or bacteria. Then we have the suppressor T cells. These cells suppress the entire immune system against the attack on the own body cells. So the function of this cells is to prevent these T cells from attacking their own body cells. And finally, memory T cells. These are previously sensitized cells, so their work is to retain the memory of this encounter with the foreign disease causing organism or the antigen, and they retain a memory for a longer time in future. Now students, we will see the mechanism of action of B lymphocytes to the antigens. B lymphocytes are sensitized directly by both the antigen as well as by the helper T cells. Activated B lymphocytes multiplies very fast to produce clones of plasma cells and memory B cells. The plasma cells produce specialized glycoproteins called as antibodies, which are circulated throughout the body fluids, that is the humor, like the blood and the lymph. Hence it is also called as humoral immunity or antibody mediated immunity. The antibody molecules may bind to a cell membrane or they may remain free. Have a look at this image here. This is your B cell. So there is a receptor on the B cell where the antigen or on the bacterium can bind. So this is one way it can get sensitized by direct counter with the antigen, or another way is the helper T cell coming in and sensitizing the B cells to undergo the cloning. So there are two ways in which your B cells can get sensitized. Now once it is sensitized either by direct contact with the antigen or with the help of the helper T cell, it forms clones where it forms the plasma cells and the memory B cells. The memory B cells obviously are going to retain a memory of this encounter with the antigen for further future use, and the plasma cells then produce the antibodies which are then circulated into our entire body fluid. Now students, the free antibodies have three main functions. First one is agglutination of the particulate matter including bacteria and viruses. The immobilized mass is then engulfed by phagocytes. Have a look at this image here. First of all, this is your antibody. This is the antigen to which it matches. This is a different antigen. For this, a different antibody will be produced. Then it will have agglutination reactions, that is the particulate antigen and antibody reactions, where if the antigen is on the cell, there is direct agglutination. Can you see here? This is your bacterium and this is your antibody. It has come and bound, forming antigen antibody complex. So it is holding the bacterium and it is thus forming an agglutination reaction where it immobilizes the bacteria. The bacteria, it cannot freely move into our system. So once it is immobilized, then it can be easily phagocytized by the phagocytes for the phagocytic cells. Then we have opsonization or coating of the bacterium to facilitate the subsequent phagocytosis by macrophages. We also have these specialized cells that can engulf the entire bacterium. For that, it has to be recognized. So this is another way, that is opsonization, that is the bacterium is coated so that a phagocytic cell can easily perform the process of phagocytosis, that is digesting the bacterial cell. Then another way in which the free antibodies can help, you can see here this antibody has helped in opsonized it and has allowed the process of phagocytosis to take place. Then the next activity that the free antibodies does is neutralization of the toxins released by the bacteria. Example, tetanus toxin. Each antibody is specific for a particular antigen. So here you can see neutralization activity happening, where again it's a little similar to your agglutination reaction where neutralization is done with the toxin. So here you do not have the bacterium but you have a toxic component of the bacterium. So this is also held and immobilized. So so in such a way that the toxin cannot have its effect on us. So this process is called as neutralization. So there are different ways in which these free antibodies help in preventing the pathogen from causing any harm to us. Then we have the antigen presenting cells. Antigen presenting cells engulf the invading pathogen and process the antigen. Then the processed antigens are presented on their own surface. Now what happens, students, is in some cases the antigen is not easily recognized as foreign. At that time, it is the work of these antigen presenting cells to engulf the pathogen, process it, and then present it as an antigen to our T cells. Have a look at this image here. We have this as your immature T cell. This is the antigen presenting cell. It has processed the incoming antigen and it has sensitized the T cell. So then the T cells gets activated and forms into the mature helper T cell and the mature cytotoxic T cells. So you can clearly see this process here also, where you can see you either have the pathogen or the vaccine antigen. So this is a similar process that happens when we are given vaccines. So it is either a component of the pathogen or it could be the pathogen itself in case of an encounter, so which is not being recognized by our lymphocytes. So at this time you can see it is taken up by the APC, it is broken down, and this step is called as antigen processing. Once it is processed, then there is a step called as antigen presentation. Now in antigen presentation, this antigens can be directly presented to the T cells. Here this is the cytotoxic T cell. So this cytotoxic T cell will perform its functions of neutralizing the antigen, and the another way is when the antigen is presented to the helper T cell, the helper T cell also goes and sensitizes the B cell. So there is B cell activation, and this gives rise to the production of memory B cell and the antibody producing plasma cell. So this is how the entire mechanism of the immune system works because of the help provided by the antigen presenting cell. So these cells are able to deliver a stimulatory signal that is necessary for the activation of helper T cells. So that's how they play an important role in activations of T cells. Thank you, students, for participating in this lecture. See you soon again with the next topic. Till then, take care, be safe, and keep learning.